just wanted to make a quick video to give you some instructions for today's activity. It's pretty straightforward, pretty simple, but without me there to walk you through it, I wanted to make sure that I did my best to make it as clear as possible. So what you're going to do today is you are going to decode DNA. So you're basically going to go through the process of transcription and translation as a bit of an activity. So what I wanted to do is I wanted to start and just walk through some of the key ideas here. So the first thing is that the DNA is on the table, all right? So one of the lab benches, probably lab bench number 1, the DNA will be on the table, and the DNA never moves. So that means that you do not move any of the cards that are on that table away from that table. And that's because DNA stays in the nucleus, and so if we want to access our DNA, we need to bring things to it. So what you're going to do is you're going to work in groups, and the groups are where you found this video on Google Classroom, okay? Each group, each group is then going to go through these steps. Somebody from the group is going to go over to the DNA, and they are going to transcribe it. So hopefully that word transcription means something to you. But what you're going to do when we say transcribe it, if I have DNA that is CAGCTT, then when I transcribe that, I am going to turn that DNA into GU, because remember, we're making RNA in transcription, and there is no T. GUCGAA. So somebody's going to go over to the DNA. You can transcribe it on the spot. Remember, don't touch the DNA cards. Don't take them away. Then you're going to bring this now messenger RNA strand back to your group, and you are going to divide it into groups of 3. So if you remember, when you have groups of 3, they are called codons. Each group of 3 is 1 codon. So once you have them broken into groups of 3, you are then going to transcribe these codons into anticodons. So my codons were GUC and GAA. So what I'm going to do now is I am going to basically find the complement again. So G becomes C, U becomes A, C becomes G. G will become C, A will become U, A will become U. So once you've done this, now keep in mind, you transcribe them at the DNA, then you break them into codons. Then those codons, you basically transcribe them again. You find what is called the complement. So the complementary base. And you will then go, you will find these on cards that are spread on the other 5 tables in the room. Now, this is very important. Don't move those cards either. You can walk over to them, look at them, find the one you're looking for, and record the word. So what do I mean by that? So when you find the cards, on the cards, there will be one of these codons, or anticodons, rather. So let's say that there's AUG. Well, on the other side of AUG, there will be a word. This is not the word for AUG. I was just making an example. You will record the word. Basically, this index card, which you will find on the other tables, represents transfer RNA. On one side of the transfer RNA, we have an anticodon, and on the other side of the transfer RNA, we have an amino acid. Once you find all the amino acids for your anticodons, you'll have a sentence. You have to find a minimum of 15 sentences. You can find more than that. Potentially, you'll get some extra credit. those are the instructions for this activity.